This video demonstrates a procedure to construct a Headstage pre-amplifier system which simultaneously records and stimulates chronically implanted electrodes in awake behaving animals. First, a printed circuit board is designed for a Headstage with a targeted number of recording and stimulating channels. Two examples of source-follower circuits are shown. The electronic components are then soldered to the circuit board, and the Headstage and tether cable connectors are constructed to provide an interface between the Headstage and the animal's implant. With no loose connections in the system, reliable local field potential recordings can be obtained from the rat hippocampus.
The main advantage of this technique over existing methods, like purchasing a commercially available Headstage amplifier system, is that this method can be thousands of dollars cheaper and easily customized for specific stimulation and recording experiments.
Because this method allows simultaneous recording and stimulation of the behaving brain, it can help identify and control neural mechanisms of cognition. In turn, these methods may ultimately help to improve function and treat brain disorders in people.
The low-cost Headstage pre-amplifier system featured in this video allows simultaneous neural recording and stimulation and can be made waterproof for use with swimming animals. A completed system consists of the following components: a computer-designed integrated circuit board with 4 recording channels and 2 stimulation channels. Gold pins relay brain signals to the Headstage and stimulation signals to the brain via separate connectors on the animal's Headstage. A tether cable and connector relays the recording and stimulation channels through a slip ring commutator to a computerized physiology workstation.